This biochemical test is collectively just referred to as the IMViC test. What the IMViC test is a series of mainly 4 different tests with some subparts to it. The I stands for the indole, M is for the methyl red, the VI is Voges-Prosker, and the C stands for citrate. The media that you will be using varies depending on which part of the test you're running. For the indole, you're going to use the SIM agar. For the methyl red, you use the MRVP broth. You also use MRVP broth for the Voges-Prosker, though heads up, you cannot use the same tube to run both tests, so you need to have 2 separate tubes of the MRVP broth. And then the citrate, we use what's known as a Simmons citrate agar. So if we look at these each separately, the indole test, we will use the SIM agar. The SIM actually you can test for hydrogen sulfide, indole, and motility. What you do is this is in a tube. It's a solid agar. So you take your needle and you just stab. It's not a slant. It's a deep. And so you would take your bacteria using your aseptic technique. You would inoculate. Take the needle and just straight down stab it and straight up. Put it in the incubator, and then after 24 hours, you're going to run this test. You're going to add the indole reagent, which is usually a Kovacs reagent. You add about 5 drops. Now, if you mistakenly add 6 or 7 and only put 4, it's not a big deal. You're going to end up with a little bit of a layer of this liquid on top of the Sims agar. If it turns red, that is positive for indole. If it remains colorless or kind of a pale yellow color to it, that is a negative for the indole. Using that same SIM tube, you look at it to test for hydrogen sulfide production, H2S. This is very easy to do because if the bacteria was able to produce H2S, it would turn it black. So if there's a black present, that is positive for hydrogen sulfide. If there's no black, then that's a negative. In terms of looking at motility, once again, you're using the same tube, so you're getting 3 tests from it. You look along the stab line. If the growth extends well beyond that stab line, then that means that it's positive for motility. If the growth is only right along that initial stab line, then it is going to be negative for motility. So here is a SIM tube. So as you can see, this is agar. There is black along the stab line, so that means that it is positive for hydrogen sulfide. The indole reagent or the Kovacs reagent has already been added. You can see it is that pinkish red color, so it is positive for indole. In the motility, the growth is only, we used a loop to inoculate, which is why it's a little bit fatter along here. But the growth did not extend beyond the stab line, so this particular organism is negative for motility. If it was positive motility, because it produces the hydrogen sulfide, this entire tube would be completely black. So that is the indole test with the side test of both hydrogen sulfide and motility. Now moving on to the methyl red. Methyl red, this is a broth. You would have inoculated it after the incubation time. You add 5 drops of methyl red to the first tube. If it turns red, it's positive. If it's colorless or yellow color, it is negative. So this is an example. You can see it's reddish along the top. That is a positive for methyl red. In your second MRVP broth, you would have inoculated, and you are now, after incubation, going to test using the Voges-Prosker test. For this one, it requires 2 separate reagents. And once again, you cannot use the same tube that you put the methyl red reagent in. So what you will do is you will add about 10 drops of the Voges-Prosker A, or we just abbreviate it as a VPA reagent. Then you add 10 drops of the VPB reagent. For the indole, the methyl red, and the VP, when you're adding these reagents on top of the broth, don't mix it in. Just let it sit on top. Just add the drops, let it stay on top. So don't mix it. So for this one, once again, you've added 10 drops of VPA, then you follow that by 10 drops of VPB. Look at the color. If it's red, it's positive. If it's colorless or a pale yellow, then it is negative for the VP. This is a MRVP tube that we've already added the reagents for the VP, and as you can see, there is no red color, therefore it is a negative for the VP. And then the citrate. You're using a Simmons citrate tube where citrate is added as a sole carbon source to see if the organism can break down the citrate. It is a slant, so you would have inoculated it after the inoculation period. Look at it. Now, uninoculated, this media is green. If it turns blue, that means it is positive for citrate utilization. If it stayed green, then that means it's negative. So on this tube, the very bottom, you can see it is still slightly green, but you can see it's this very pretty blue coloring. So this would be positive for citrate. Once again, all of these tests you would be recording, and then the results of them can help you to identify an unknown bacteria.